I hope all of you have the same sense that I do, that this has been a pretty remarkable 2 days. A lot of sharing of a lot of information, a lot of learning, and also a sense of complexity. It goes without saying, but maybe needs to be said, that we're living in a time of remarkable scientific change. Day to day, I am reclassifying and rethinking about diseases and conditions that I was taught about in medical school back in the 1960s, which were quite wrong, and reassessing my understanding of the basis of the diseases. And with that assessment of understanding of differences in etiology and cause of conditions, thinking about very radically different ways in which we treat disease. I just want to share with you a few bits of data from FDA to give you at least my sense of where the whole issue of diagnostics and therapeutics, of personalized medicine, of rare diseases is going, because it is going really, really rapidly. And as regulatory agencies, we indeed need to stay ahead of the scientific curve in partnership with our colleagues in academia and with those who are actually discovering, testing, and manufacturing products. 2011 was an amazing year for the FDA. We approved 30 new molecular entities. That's the highest in a whole series of years. But looking under it in those numbers is the most interesting part. More than half were novel compounds, first in class, things never imagined before. More than half were defined as rare and orphan diseases. This is a huge temporal increase in the percentage of compounds coming to us defined in the orphan space. And the rare and orphan disease space included both traditional things that I have been able to recognize as syndromes, but also recognizing the subdivision of diseases into biologically relevant, clinically meaningful subtypes that indeed fit under our Orphan Drug Act. The 15 of the compounds of the 30 received priority status. Now, in the US, we do a lot of talking with sponsors early, end of IND, end of phase 2, a lot of discussions about study design. Of those 15 priority compounds, all were approved, 100% first round review. That beats everything that we've talked about before. We've argued that understanding disease etiology, understanding biology, and personalizing therapeutics would also speed drug development. It has in spades. We have 3 programs, total development time between 4 and 6 years as opposed to 10 and 12 years. 3 compounds reviewed internally in 3 months, less than half the normal expedited review time. Why? Very simple, the science. The science was so good, so compelling, that there really wasn't much of an issue. And what do you see when you start getting compounds that are highly targeted? This hasn't been talked about much here. You get increased effect size, and you get increased percentage of patients responding. You don't need to do a 7,000 patient phase 3 study to show drug effect in 55%, placebo response 40%, and in my eyes, therefore, drug attributable response of 15%. We're seeing drugs that work in 85, 90, 90% of patients with effect sizes that, with apologies to our statisticians, you don't need a statistical analysis to see the effects, because the effect sizes are so large, because we are attacking molecular etiology. I'm very optimistic about this. I've been a physician for 35 years. I've struggled using drugs based on population data that I know are not working in my individual patients. And I'm now using medicines that I have increased confidence of because, in fact, they are targeted. It leads for all of us huge challenges of working out diagnostics and therapeutics. I'm lucky at FDA because my directorate includes the Center for Drugs, the Center for Biologics, and the Center for Devices and Radiologic Health, which includes our in vitro diagnostics groups. And our folks enjoy working together and talking to each other about complex issues and pushing the limits in terms of study design and clinical trials that match the molecular side, understanding both phenotype, clinical manifestations, clinically meaningful outcomes for our patients, as well as being guided increasingly by more and more sophisticated biomarkers, and also recognizing, which our colleagues at the National Institutes of Health often don't understand, what validity means. Both analytical validity of the tests that we use, clinical validity and the associations between a given marker and a clinical outcome. To many of my basic science colleagues, validity is really what I consider discovery. And there's a big gap between a reagent and an actual medicine that can be used in real patients in real time. Huge challenges for us all. It is not going to stop. Companion diagnostics, we've had a discussion of, I think they're going to disappear as an entity within 10 years. If we all know our genotype and have whole genome sequencing done, which some would argue is going to happen in 5 years, I would argue probably 15 years, companion diagnostic, it doesn't mean anything anymore if we actually know our genotypes, just like children with cystic fibrosis know their genotype at the time of diagnosis, so that you don't have to do a genotype to select specific medicines. Things are going to happen fast. And because of the complexity, my one plea for all of us, we are all in this together. This is the major time when international collaboration and all of us who see different aspects and different parts of the problem and solution need to work together. And that's why it's particularly wonderful that we've had this get together and to share different ideas and different vantage points and different viewpoints because we have a lot of problem solving to do. Although there are those who are spending almost all their time critiquing medicine, critiquing science, we need to be in a problem-solving mode, truly solving problems together, because our job is taking care of patients and increasing the public health, both.